All right. Hello, everyone. I see attendees are piling into the webinar now, so we'll give everybody a minute or two. Welcome, everyone. I see folks are still entering the room for the research update on the IQCB1 NPHP5-associated retinal dystrophy. Thank you for joining. Just going to give everybody here another minute. Folks are still coming in. All right, wonderful. All right, let's go ahead and get started. I'd like to once again welcome all of you to the research update for IQCB1 NPHP5 community. My name is Laura Manfrey. I'm a co-founder and the board chair of Sophia Sees Hope, which is a patient advocacy organization dedicated to supporting the LCA community. So we do a few things. We raise funds for research, which we do in very close partnership with the Foundation Fighting Blindness. We also support with funding their My Retina Tracker program, funding and awareness, I should say. It's a very important program and patient registry. And then we also have our own programs where we provide support and education to all of those affected with LCA, regardless of genetic variants, as well as those who maybe only have a clinical diagnosis and not a genetic diagnosis. Quick touch on some of the programs in case some of you are not familiar with us again. So we have a Family Connections program. We have a team of ambassadors as well that help us to expand our reach globally. We have a newsletter called Seeing Hope, which we're very happy to mail anywhere. There's also a digital version. You can sign up on our website. And we are hosting a monthly series now called Let's Chat About. So we had one this week. Every month we cover a different topic in research or regulatory or industry. So we invite you to join those as well. Pre-COVID, we had hosted a couple of family conferences, and we hope to be able to return to that again in a face-to-face, in-person format soon, perhaps next year or the year after. And then, of course, we're constantly sharing stories, resources, etc., creating, generating awareness with various campaigns, and always looking to hear from the patient community so we can support your needs as well. So that's a little bit about us. Some housekeeping next. So this is a webinar format. Your cameras are disabled. So if you're worried about being seen, don't worry. Cameras are turned off for participants. Your microphones are also muted. This means that if you do wish to ask questions, please use the question manager, and the question manager is available from the bottom of your Zoom panel there. And Lee-Isla Kenny, who is here, will be helping to support us with questions. You can also raise your hand, and we will send instructions to take you off mute, to unmute yourself, and then be able to ask a question verbally. Finally, this session is being recorded. So in case anybody missed it and would like to capture it, or you'd like to listen again, we will make that recording available. All right, so our objective today is really to engage you, our IQCB1 NPHP5 community, as we seek to advance treatment for ourselves and our loved ones. We're going to start first by providing some background on a scientific advancement workshop that took place last week. So just 1 week ago, it's pretty fresh. Very excited about that. And then Todd Durham, who's here from the Foundation Fighting Blindness, will walk through the state of the community with the survey results that many of you participated in helping us gather that data. So thank you. Next, Dr. Amy Lassiter will walk through some of the research highlights from the advancement workshop. We'll have time for Q&A and then some closing remarks. In terms of what to expect from today, so really, really important from the next hour, definitely expect highlights from the workshop. We have some exciting news to share. A summary of the research, and of course an opportunity to be heard. So absolutely raise your hand, pop any questions you have into the question manager. What not to expect? I know this is very close to, for many of us, it's very personal. It's very personal for me. This is actually my daughter's genetic variant, this particular one. So just want to make sure that we are setting the expectation here as well. This is not going to be a deep dive into the science, which is rather complex. There are also no easy answers, right? And there's no quick or fast solution. So just wanted to set that expectation up front, even as we are very excited to share what the news that we have with you. So a little bit of background on the scientific advancement workshop that took place last week. That objective was to identify actionable steps to advance the development of treatment for those living with IQCB1 NPHP5-associated retinal disease. And we did this by bringing together the top research experts in the field working on this variant, or in ophthalmology and gene research, right? Having them share their information, ask questions, and help us to identify gaps and next steps. We also did this by sharing the information from the patient survey as well, and sharing some patient perspectives. So this slide, I Googled when I was making this PowerPoint. I looked up celebration images and found the zebra, which represents rare disease. It's a piñata and she's smashing it open. And it was the perfect image for this, because with the workshops, we weren't really sure what we were going to get when we bring all these top experts with different opinions, different approaches into the same room. But the good news is who we got was really pretty exciting, frankly. We hit the piñata. We had over 40 experts in the room, I think even over 50 for this particular workshop, which is very exciting. It was virtual, it was 5 hours, and it really fostered this, we saw the collaboration happening amongst all these various stakeholders. We were able to build that framework and platform for knowledge sharing, and really just broaden the circle of research awareness around our gene, right? Very, very important too, as folks came together for the survey and the results that we presented, it really just demonstrated that we have an engaged community. We have folks who have this gene identified, and that's really, really important. So again, thanks to all who participated with that. So what is still a work in progress, obviously, are exactly what those specific next steps are and the priority actions to take the research forward. But we'll share a little bit about that coming soon. So I am going to pause here to see if there are any questions or comments before we move on to the next section, at which point I will introduce Todd Durham from the Foundation Fighting Blindness. All right. So thank you all again. We're excited to share these updates. And Todd, I will let you introduce yourself and walk us through the survey.
Thank you, Laura. Let me just take one moment here to share my screen. Yeah, hello, my name is Todd Durham. I'm the Vice President of Clinical and Outcomes Research at the Foundation Fighting Blindness. I think many of you know quite a bit about us already, but I'll give a brief introduction for those who aren't. Our mission is to drive the research that would lead to preventions, treatments, and vision restoration for the degenerative retinal diseases, including macular degeneration, retinitis pigmentosa, and Usher syndrome. And together, these affect more than 10 million Americans and millions more around the world. The foundation was founded in 1971, and today we're the leading private funder of retinal disease research. And this is an overview of our organization, which you can also find more about on our website. At our scientific advancement workshop last week, we presented the results of a survey, which many of you will have completed in advance. Really, what the purpose of this survey is to bring to this workshop the voices of those families and individuals who are affected by IQCB1. And since I presented to the research community last week, I thought you may also enjoy hearing what they heard. We developed a survey of affected individuals and their caregivers, and we wanted to gather information from this community to share with the researchers. We recruited for the survey through My Retina Tracker Registry, which I'm responsible for at the foundation through an e-blast or email recruitment. And also Sophia Sees Hope and other groups helped us circulate the link for the survey. It was self-administered through SurveyMonkey. And what I'm presenting today are the responses from 16 respondents. 13 identified themselves as caregivers and 3 individuals as themselves affected with IQCB1 associated retinal dystrophy, for a total of 20 affected individuals represented in the survey. And 11 of the 16 respondents have a profile in My Retina Tracker Registry. So thank you very much for your ongoing support of the foundation through that effort. On this slide is a little bit of information what we learned about the diagnostic journey. We asked one question: what age was the affected individual first diagnosed clinically? On this table is a set of summary statistics. It shows the average age was 4.7 or 5 years of age. The median was 1. So this means that half of the individuals about whom the diagnosis was reported in this survey had their clinical diagnosis within the first year of life, and this is consistent with most of the reports we hear about IQCB1. We also ask a separate question about what age was the affected individual first diagnosed genetically, and the summary statistics here would show that there was a gap between the initial clinical diagnosis and the genetic confirmation of it, with a median age of 6 and a mean of 15. We ask about the current best corrected visual acuity, and what's shown in this table is that 7 out of the 16 respondents reported that the best corrected visual acuity was light perception or no light perception. So that was the most frequently reported category, followed by 20 over 200 to 20 over 400. And for context, think about how this may relate to others that you may know or your own experience. The median age of individuals in the sample is 16 years of age. And what I'm showing on this slide is a set of 6 stacked bar charts. We asked about visual symptoms experienced by the affected individual. And for each of these 6 symptoms, one could rate the severity as not applicable, mild, medium, or severe. On the left-hand side, the stacked bar chart shows that in yellow and orange is about 20% of the individuals in this sample reported either moderate, medium, or severe nystagmus, leaving the remainder 80-some% who either did not experience this symptom or only mild symptom. And then contrast that on the right-hand side, where you see that 90% of individuals in this sample reported experience either moderate or severe night blindness. And this is also consistent with what we see in reports of the clinical manifestations of IQCB1. We also ask about other related conditions. The question was, are there other symptoms or physical health conditions, eye-related or otherwise, that are related to the individual's diagnosed genetic variant? The most common response was about kidney failure or senior Loken syndrome, with 8 responses. 5 individuals had no other related condition, and 2 reported a potential link to autism, although the relationship isn't really known. We also ask think about yours or your affected individual's condition. What worries you the most? And the most commonly reported worry here is reliance on others or loss of independence, followed by kidney failure, progression to blindness, and employment discrimination or finding work, with 2 responses. One of the more powerful ways that we can learn about the experience is not to code the answers as a number or a category from the survey, but is to represent your experience in the words you provide us. So here is just an example on this slide of what was presented last week. We ask, please feel free to elaborate on the impact to your family overall, including family members who are not directly affected with the disease. So one person said depression, sadness, complexity to organize our life. Another said, since being unable to drive, my career prospects have been very limited. As a husband and father, my ability to earn a livelihood, socialize, or participate in hobbies has been severely limited. I cannot go anywhere without being driven. And another one I'll just read here is going out as a family, schoolwork, work schedules, health, shopping, playing outside, autonomy, and independence. We also want to take an opportunity on the occasion of these advancement workshops to think about interest and knowledge about clinical trials. We ask, would you consider participating in or supporting your family members' participation in a clinical trial to treat your form of blindness? Of the 12 responses to this questions, 9 individuals said yes, and 3 were unsure at this time. We also then ask, what are the top 1 to 3 questions you have about being in a trial, and what information would you need or want to know before participating? It's not surprising that the most common category here was general risks, like is it safe? Followed by, are there any risks to the kidney? What are the time commitments involved, and will there be pain or discomfort? And how about risk to my remaining vision? And then there were a handful of other responses there as well. And also, we ask about what were individuals' motivations for participating in a clinical trial. So some examples are to cure and help people with independence, bring some usable vision that will promote a more independent life, an improved visual field, and to help others. So it's not uncommon for us to see, in addition to hope for your own improvement in your health, your vision, also the factor of helping others is apparent in all these survey responses. And then we left one more opportunity at the end of the survey to ask, is there anything else you wanted to share with the researchers? And you see the responses here. Is there any research into this would hugely benefit our and our other families? We hope research continues to move forward. Another respondent said, time is vision. Please hurry for IQCB1, give us hope. Another said, I'm so happy to hear that there is ongoing interest in this particular genetic variant. Please keep working in this area. And finally, we've met plenty of people with this and other conditions causing blindness, and the quality of life and independence for individuals who at one time or still now have usable vision compared to those completely blind since birth is enormous. So I think we see in a couple of responses there the urgency and the need to preserve or restore vision that's possible. So I want to thank everyone for responding to this survey, and also remind you, if you saw the announcement or would still like to respond to the survey, we're going to keep it open through the end of this week, at which point it'll be closed, and I will do a final analysis of this total survey, which will be presented in the final report from the Scientific Advancement Workshop, and ultimately be made available to the community at large. So with that, I will wrap then, and then I will turn this over to my colleague, Amy Laster.
Mute. All right, great. Great. Thanks, Todd. Good day, everyone. Again, I am Amy Laster, the Vice President of Science and Awards Program with the Foundation Fighting Blindness. Todd, are you going to put the slide back up?
I told you I would, didn't I, Amy? I'm sorry. Okay, just a moment.
So on this slide, it's a text-based graphic that's depicting the development path from research to cure. We're getting there.
Do you see it now?
There it is. Yes, it's there.
Okay.
Thank you. So as we begin to think about achieving therapies for IQCB1, NPHP5, there is a development pipeline that begins with first understanding the gene and the pathological and disease-causing mutations or variants. And so from here, we can assess the impact on the associated proteins, in this case, nephrocysteine, and its related partners, as well as their function. So this allows us to establish really the fundamental knowledge that gives us understanding of the disease, helps us to think about best disease models, the course of the disease, and potentially some ways to treat the disease. So for IQCB1, the research is really at the early stage of translational development or translational research. So that's kind of here. There's a yellow bar kind of at the bottom of this graphic for translational research. So we can kind of think about it being, again, towards the beginning of that developmental stage. From last week's workshop, just kind of touching again on the high level highlights, Dr. Gustavo Aguirre, who did an awesome job presenting what his laboratory is doing, and in particular, the results of his proof of concept gene therapy study in canines or dogs, which will soon be published in a peer-reviewed journal. There is a preprint that's openly available to the public. There's a journal platform called bioRxiv, and so we can make sure that everyone have that information if you want to take a look at that. So the research showed that the photoreceptors in the treated areas, that they reform their structure. And there was an improvement in function as measured by ERGs. So the dogs in the studies, they were sent through an obstacle course in under two conditions, in dim light as well as bright light. And when they were navigating with their treated eye, the dogs had faster travel times and fewer collisions than when they were navigating using their untreated eye. So this was really good outcomes for this proof of concept study, very promising, and we're excited about these results. I do also want to mention that in addition, there's ongoing clinical studies that are characterizing human cohorts, suggesting that the structure of the central retina or the macula, it's preserved for many years, despite having fairly significant loss of vision. So this, if you will, kind of dissociation between the structure and function suggests that a gene therapy targeted at the central retina not only could work, but has a good potential to restore lost vision. So we build on this proof of concept and clinical knowledge. Again, just kind of looking at our graphic here towards translating these laboratory-based research concepts into clinical trials for patients. First, there needs to be time devoted to designing the clinical trials for our target population. We'll go through the clinical stages of identifying what are the relevant endpoints and outcomes to measure the effectiveness of the gene therapy. And then from here, then really clinical translation, it moves through those phases of clinical trials that will test the safety and effectiveness of a therapy. So we're really encouraged, again, where things are now. We've learned last week from Dr. Aguirre that he and his colleague, Dr. William Beltran, they are scientific co-founders of a company, Luxviat Therapeutics, and this would really position them to advance the gene therapy into clinical trials. And further to this, the foundation will continue to do all that we can to accelerate any clinical development. So again, this is just very high level outcomes, but the key points really of last week's workshop. So I'm going to stop there now. See if we want to dive into next steps. any questions? In the Q&A chat.
Amy, we have one question in the Q&A at the moment. It's from David H. I've spent most of my life incorrectly diagnosed with RP. I did not learn I had LCA until I had genetic testing provided by Spark. Do you think this is common, and how can it be addressed?
Todd, you want to take that?
Yeah, thanks for the question, David. My impression is your story is not very unusual at all. I think the reason for this is the clinical manifestations, or what many ophthalmologists can observe when they do an exam, can overlap quite a bit from various conditions. I think ultimately, at the end of the day, the fact that you got a genetic diagnosis is most informative for you, and that's our hope, that there will be more access to genetic testing. And if you're not aware, we do have a genetic testing program that offers this genetic test to individuals at no cost to themselves. That program has been available since 2017. So we see this as a challenge for the community as a whole. And just to reassure you, I don't think your story is very unusual at all, unfortunately, but hopefully access genetic testing going forward is going to make that a less frequent story.
I think we have a hand raised.
Let's see.
I don't know if you can hear me now.
We can hear
Yes.
you.
Oh, awesome. Okay. Hi. My son is 4 years old and got diagnosed with ICBQ1 at 1.5 years. And he does have something present in his kidney, so he has nephronephrosis. So we're being followed by the hospital for sick kids, and they've been, like, Dr. Vincent and the whole team has been phenomenal. My question is when we got tested, they were under the impression that both parents would have had it in our cases, but in my son's case, only one parent has it. And then, my son ended up getting the gene. Have you seen that happen with other patients? And does it matter or not matter at all when it comes to the disease progression? Because we're told he doesn't have LCA, but he has ICBQ1. I don't know if I confused you there, but I guess what I'm asking is do you have other patients with similar kind of diagnosis where they don't have LCA but do have ICBQ1? And does the progression kind of follow a different or similar path for all of them?
Yeah, thank you for that question. So typically IQCB1 related disease, it's a recessive disease. And so that means that there's a mutation in 2 gene alleles, so one from each parent. And IQCB1, it can be syndromic. And so there is the eye disease as well as a renal dysfunction as well, or it could just be exclusively a non-syndromic and only an eye disease or an LCA. So that's not uncommon. It depends on the specific variant in the gene that's inherited. And so a parent could be a carrier of the particular variant, but does not express any clinical phenotypes.
Okay, yeah, because that's exactly what has happened in our case. And so I was just wondering if other patients, if you've seen other patients like that, and has I think in the survey it seemed like most of the respondents or caregivers have noted that by 16 they've been affected by night blindness 90% and that type of thing. I guess what I'm trying to do is just I'm trying to run a mile a minute when I shouldn't. And I'm trying to just understand my son's path based on other patients' paths. Like, he's 4 now, and when is he gonna, I hope to God that he doesn't ever see any of these things and there's a cure and treatment for everyone. But if he does, what is the sort of the progression of of the disease relative to all of his other peers that have faced it.
Yeah. Thank you.
We have a few more questions in the chat box. We have one from Henry Kornman. Could you repeat the name of the company that will sponsor the clinical trial? And then he also asks, is gene therapy the only path currently for IQCB1? When can we expect a first clinical trial?
All good questions. The company by Dr. Beltran and Dr. Aguirre is Lux Fiat, and I did type it in the chat just in case that will help. Oftentimes these very early companies, you won't be able to find anything on them. This is just their announcement of this, that they are forming and working toward this. So toward other treatment therapies, we did explore that with our experts last week. And in terms of what has advanced, there are some potential, maybe small molecules. There is some interest in exploring that, but there has not been anything that's at a proof of concept as was shown for the dog studies and the gene therapy that's currently out there. As I mentioned, when we are at a translational research stage, as these dog studies are, there are a number of next steps with regards to doing some safety studies and some toxicity studies on the gene therapy before there are those conversations about getting acceptance to move into a clinical trial. So really, just honestly, at this time, we don't have a timeline for when the clinical trials will begin. Thank you.
Okay, we have one more question from Christina Defina. Does any of the research have potential to stop the kidney failure part of this disease, or is it only concentrating on the eyesight?
Yeah, good question, Christina. So this particular strategy, it is a gene therapy, which would target restoring function of the IQCB1 protein in the eye. So this would be specific to the eye.
Okay, and then our final question is from David Friedrich. The canine trial was made known 4+ years ago. Did the researchers' results signify the conclusion of that phase of their work and moving on to the next phase? Are there financial hurdles that have slowed their progress?
Yes, again, good question. So these trials, you start them and there are even stages. So for example, you may do preliminary studies to simply test what happens if we were to treat with a gene therapy. Then you have to understand, are there better components of the gene therapy system that would result in more effectiveness of the treatment? And then you want to know, so what happens after 3 months? What happens after 6 months? So sort of immediate short term. And then you continue to ask those questions. Well, what about long-term data? And so while this initiated 4 years ago, the outcomes that Dr. Aguirre were able to provide last week, and again, that will be published, will really kind of button up his proof of concept study with the long-term data, which was has only recently been available. So it's more of a just timing to take to get here, and not that things had slowed down. They clearly have expressed to us that they have been actively working on this program, and this is a program that they anticipate continuing to work on.
And Amy, if I might add too, Dr. Aguirre also told us the paper that's available currently in the pre-press bioarchive now he's still under final consideration and final editing with a peer-reviewed journal. So I imagine some of their progress is sort of waiting on the validation that comes from a publication in the peer-reviewed scientific literature. So I imagine that's part of it, but the research itself is already available. But and the other thing I might add is there's likely, the way these things happen is likely much activity going on behind the scenes. It's not publicly known and often not known by the foundation, because many of these organizations have much at stake to move the therapies forward. As you can imagine, these are, some significant interest here. So that maybe the study concluded years ago doesn't mean that they haven't been actively working with such intriguing and exciting results from this non-clinical model. I think there's a lot of enthusiasm about this approach.
We have 2 more questions in the chat box. There's a follow-up question from Christina. Do you know of any research being done to correct the function of the gene for the kidney?
Christina, thank you for your question. I haven't delved into specific laboratories on this, but I do know that there is a large body of work that is being conducted on the renal function of it. In fact, that is the tissue that was first identified as being affected by mutations in the NPHP5 or IQCB1. There is a large online based registry for the kidney portion of it. So if you want to follow up, Laura and Todd's email will be presented at the end. Then I'm happy to send you some, kind of information that I have found, but I don't have specifics on the research on the kidney function.
Okay, and then we have a question from Jassy Gill. How long does it take typically from research to treatment?
Todd, do you want to talk about the clinical trial process?
Well, let me go back here. Maybe I could go back a slide. I think there's a couple elements to this question. One is this process from the basic to the translational research part can take decades. And as I understand it, the foundation has funded a number of these researchers for many years, so that going from the basic understanding of the gene, the proteins that are expressed and the function of those proteins and their role in the disease pathophysiology can take decades. Boy, it's kind of hard to generalize a timeline. I imagine if there were company positioned ready to develop put together what's called an investigational new drug application, that is to seek authorization to treat humans, it could take a year or two to initiate, but oftentimes those the sponsors of those programs will time their IND application so they're hopefully within 1 year of going into humans. We're kind of in this unknown zone now to know where the state of this IQCB1 technology is. Up into that IND, there's a step that's required to do toxicology studies to make sure that it would be okay and reasonably safe to proceed in a human. And we don't know exactly what the stage of those studies are at the moment, but those will be prerequisites in addition to this. Non-clinical model in the dog research is a key part of this, which is the demonstration there is a potential for benefit in humans, and then that has to be balanced on the safety side by looking at potential issues that might be envisioned once humans are treated with the gene therapy. So it's hard to say. It is uncertain until we see the IND to know exactly when the first human could be treated with the gene therapy. Unfortunately, it's one of those unknowns to us at the moment. But they've very much ticked off a major accomplishment and milestone here on the path to the IND in the first human study in this proof of concept model. And Amy, I don't know if you'd add any more to that.
No, that's very a good assessment. Even you know, when we think about things that are sort of in our translational stage, our early translational stage, I'll just say projects in general that we may support. They could be at this stage, as Todd said, you know, up to 10 years, but typically it's in the 5-year range. And it just really depends on what's already known. I mean, one thing that is to be leveraged is that there have been successful gene therapy trials in ophthalmology. And so there's lots of things that we have learned, and those things are certainly benefiting gene therapy and ophthalmology in terms of the amount of work and the pathway forward. So particularly in this case with Dr. Aguirre and his team, who was very instrumental in the success of the currently approved Luxturna, right? And so they're very familiar with the path to the clinic. So they're not learning all the pieces, right? They can focus on this particular disease and understand what are the key questions to ask. But in terms of the process to advance it, they're keenly aware of that. So that's just advantageous. And I understand it's still not giving you a precise timeline. But again, hopefully encouraging that we've learned about gene therapy. And so while for RPE65, you know, it may have taken somewhere like over 15 years to get there, that's clearly not what we're seeing here. So.
Mm-hmm.
Any other questions? Feel free. And thank you, Henry, for your comment.
I just wanted to add that if you are on the phone, you can press *9 to raise your hand and then *6 to unmute or mute yourself. Okay, we have one question from Nicole. You said that it took 15 years with Luxturna. How long have they been working on the gene therapy for this one?
Nicole, I'm not exactly sure when they started working on this. And your question was specific to gene therapy, so I would take it to Dr. Aguirre's work. I'm not exactly sure when they began working on this. And I think it goes back to when they identified the K9 model. And that number is not on the top of my head right now. So it's been about 5 years. We certainly hope that advancing these therapies will get faster, right? The more we know, or at least more efficient.
Yeah, Amy made a great point that when you're developing a new medicine, if you've had a product come before it, this paved the way. You don't have to address the same questions with the regulators at the end of the day about what would be acceptable, measure of success, what's the sufficiency of data. So I think the more of these we see, the more efficient the whole process is going to be. And the regulators also develop some comfort level too. Okay, Laura and Amy, I wonder if we should, okay, this one is perfect for Laura to respond to.
Yes, so thanks for this question. Is there an IQCB1 support community? Thanks for that question, Jesse. So Sophia Sees Hope supports all folks with LCA, so across all genetic variants. But we also have a resource page on our website where we list various support groups by genetic variant. And for IQCB1, there is a really great Facebook group. So I believe that's on our website resources page. We can make sure in the follow-up that we're sending a link to that Facebook group out to everyone on this call. And that's really been a very helpful source, frankly, even for me belonging to this community with my child who has that variant. I found the group and there's over 30 folks in there. So there's questions. We'll post the survey link there as well for folks who haven't taken our community survey yet to take it again so we can get all that data pulled together. But it's a really nice community there.
I'll take David's question. So yes, the foundation has been supporting Dr. Aguirre and his team through a multi-investigator grant, which is a large animal study. So we've been supporting the Pennsylvania's canine facility for a number of years for the maintaining of their canine colonies to identify additional variants within the canine colony, having therefore naturally occurring disease, which again makes for great models for testing therapies. And so any support, obviously that's given to the foundation, is to support our research programs, and that would include Dr. Aguirre's research program at Pennsylvania.
Yeah, so here's an open question too about posting the link for the preprint. Yes, we can. Because I'm driving the slides here, I can't escape from the window to go find it, but we can send out a link for the bioarchive.
And a quick note, since I am not sharing slides right now, I did just post a link to the IQCB1 Facebook group in the chat there, so everyone has that.
Okay, last call for questions, I guess. Laura, I'll turn it back over to you then.
Okay, that's great. Do you mind driving the slides?
Sure.
All right, and actually, Todd, do you want to speak to this one? I know we spoke a little bit about this since the
Sure.
faculty is publishing them.
Yeah, so what's to come after the part of the workshop is dissemination of what we learned and to memorialize that occasion. So we are at the moment preparing with the scientific writing team a document that will say who was there, what we talked about, what the key learnings were. So we are that'll take probably a couple of months to get that finished. So it'll have to be drafted. We'll ask the attendees of the workshop to review it and give us their comments, and then we'll be able to disseminate it. And if all goes well, we'll get this published in a peer-reviewed article as well. And the other part too is my colleague Amy Laster prepared a landscape document, really a high-level summary of the research that's been done on IQCB1 to date. And this landscape document will be made available and part of the summary report from the workshop itself, in addition to the survey results. Okay, Laura.
Yeah, great. So really exciting news. I hope everyone on this call is really excited just about what they heard. I know we don't have any direct answers. As I said at the beginning, we can't say there'll be a treatment in 5 years or 10 years. There's still so many things that need to happen. But honestly, this workshop last week really just crystallized and pulled together where things are. So we can see a path forward. And, it took 7 years for us just to get a diagnosis of IQCB1 for our daughter. And then we just kind of sat and waited. So to know what happened before and where we are today, it's just huge. It really is incredible progress, and I'm very encouraged. So what we can do as a patient community, I think is also very important here, right? So I would encourage everyone, if they're open to it, to please create a profile in my retina tracker. That is the registry that's managed by the Foundation Fighting Blindness. And so when clinical trials happen, or there's natural history studies, when there's news to be shared, they can look for who is affected, and they will email directly from that registry and that database there. So really, really important. Plus it helps collect information on our community, and we need to show up, right? We need to be responsible and do everything that we can to make sure we are readily available, informed, and show that we are here to receive a treatment, right? So the other thing to do, obviously, is follow the Foundation Fighting Blindness. They've got a great research blog. I mean, they're incredible experts here. Todd and Amy obviously are two of them, but there's a really great team there that's constantly following all the science and funding these research efforts that are hopefully advancing and taking us forward to treatment. And then follow Sophia Sees Hope as well. So, on the question about, is there an IQCB1 support group? I saw your chat there, not on Facebook, and I know a lot of other people aren't. So that's an opportunity then to connect really with us at Sophia Sees Hope. Again, we serve the whole LCA community, but we do have a family connections program. So we're happy to connect you with other people in that community. We publish a newsletter. So, other ways to stay in touch with us and have those connections to other folks in IQCB1. And then, if you're wondering if you can do more, sure. Like, we need to get the word out to help grow our community. So if you're working with a genetic counselor or your retinal specialist, please, by all means, help spread the word about Sophia Sees Hope and about Foundation Fighting Blindness. If they know where the patients are, and they can then share this information about my retina tracker and about Sophia Sees Hope with their communities, so that we can continue to grow our patient group here. And I think that's really key, and that's something that we can do. I was so encouraged. Before we did the scientific advancement workshops, I probably knew about 5 other people with IQCB1, right? So having gone through this, having reached out and started creating this buzz around it, I think we went from 7 people, 7 individuals with IQCB1 registered in my retina tracker. Well, now we've identified over 20, I think over 25, in fact. So, the larger our community, the more we can show up, and the more interesting, exciting this is, and the more we can really support advancing this treatment. So that's my soapbox. I will shut up. I will put a link to the survey in the chat window here as well. So let me pop that in. So if anyone hasn't been able to participate in that, please do. We will also send that out to all of the registrants via email, so that you have that there also. And I just want to say, thank you on behalf of the Foundation Fighting Blindness and Sophia Sees Hope. Thank you so much. And Todd, if you flip one slide over, oh, there we go. If you do have, other questions, there's Todd's email, there's my email, and we're here to support. Very, very encouraged and excited about what the future holds. Thank you, Leila, Todd, Amy.
Well, thank you, Laura. And I just wanted to reiterate and emphasize a point that Laura just made about the importance of community. we have opportunities to speak with companies all the time, and I spoke with a company earlier this week who noticed for a more prevalent condition than for IQCB1 that there is not an active patient engagement for this particular community, and they have identified this as a problem for them because they're thinking about, if we have a clinical trial program, what will be the source by which we are able to engage folks and get them interested in this community. So the foundation is committed to this, but we can't do it alone. We have to work with organizations like Sophia Sees Hope and others. So this needs to really be a community effort to stay engaged and get organized. So thank you. And thank you to Laura, who has done a ton of work on behalf of Sophia Sees Hope. You may not know this is not her full-time job. This is her passion, and I really appreciate working with her on this. It's been a joy, and we're really excited about what the future holds.
Thank you. Same here. Thank you, everyone. And the recording will be posted. So we will post this recording. Just give us a couple days. Thank you all.
Thank you. Bye.